In this section, we will talk about the diffusion through membranes. Up to now, we have considered general features of diffusion of solutes in water, in living tissue. However, diffusion often occurs across cellular membranes, including between intracellular and extracellular fluid compartments. For example, cellular waste products of metabolism diffuse outward from cells, whereas nutrients diffuse into cells. In both cases, the solutes must cross the plasma membrane. So what effects do membranes have on diffusion? Well, the rate at which a substance diffuses across a plasma membrane can be measured by monitoring the rate at which its intracellular concentration approaches diffusion equilibrium with its concentration in the extracellular fluid. Now, let's plot and graph. To understand this, having concentration on x-axis and the time on y-axis. The increase in intracellular concentration as a solute diffuses from a constant extracellular concentration until diffusion equilibrium is reached across the plasma membrane of a cell. As with all diffusion processes, the net flux of material across the membrane is from the region of greater concentration, which is the extracellular solution in this case, to the region of less concentration, which is the intracellular fluid. The magnitude of the net flux, that is, the rate of diffusion represented by J, is directly proportional to the difference in concentration across the membrane, CaO minus Ci, where O and I stand for concentrations outside and inside the cell. The surface area of the membrane, represented by A, and the membrane permeability coefficient, represented by P as described by a modified form of Fick's first law of diffusion applied to biological membranes. The numerical value of the permeability coefficient is an experimentally determined number for a particular type of molecule at a given temperature. It reflects the ease with which the molecule is able to move through a given membrane. In other words, the greater the permeability coefficient, the faster the net flux across the membrane for any given concentration difference and membrane surface area. Depending on the magnitude of their permeability coefficients, molecules typically diffuse 1,000 to 1,000,000 times slower through membranes than through a water layer of equal thickness. Membranes, therefore, act as barriers that considerably slow the diffusion of molecules across their surfaces. The major factor limiting diffusion across a membrane is its chemical composition, namely the hydrophobic interior of its lipid bilayer. So, let's understand diffusion through the lipid bilayer. When the permeability coefficients of different organic molecules are examined in relation to their molecular structures, a correlation emerges, whereas most polar molecules diffuse into cells very slowly or not at all. Non-polar molecules diffuse much more rapidly across plasma membranes. That is, they have large permeability coefficients. But how non-polar molecules have large permeability coefficients? Let's discuss this in detail. Well, this is plasma membrane, as we know that. Head are polar, while tails are nonpolar. So the reason is that nonpolar molecules can dissolve in the nonpolar regions of the membrane, occupied by the fatty acid chains of the membrane phospholipids. In contrast, polar molecules have a much lower solubility in the membrane lipids, increasing the lipid solubility of a substance by decreasing the number of polar or ionized groups it contains will increase the number of molecules dissolved in the membrane lipids. This will increase the flux of the substance across the membrane. Oxygen, carbon dioxide, fatty acids, and steroid hormones are examples of nonpolar molecules that diffuse rapidly through the lipid portions of membranes. Most of the organic molecules that make up the intermediate stages of the various metabolic pathways are ionized or polar molecules, often containing an ionized phosphate group. Therefore, they have a low solubility in the lipid bilayer. Most of these substances are retained within cells and organelles because they cannot diffuse across the lipid bilayer of membranes unless the membrane contains special proteins, such as ion channels, as we see next. This is an excellent example of the general principle of physiology that physiological processes are dictated by the laws of chemistry and physics. Now, moving towards diffusion of ions through ion channels. From natural lipid bilayers, ions such as sodium, potassium, chloride, and calcium diffuse across plasma membranes at much faster rates than would be predicted from their very low solubility in membrane lipids. Also, different cells have quite different permeabilities to these ions, whereas nonpolar substances have similar permeabilities in nearly all cells. Moreover, artificial lipid bilayers containing no protein are practically impermeable to these ions. This indicates that the protein component of the membrane is responsible for these permeability differences. An important characteristic of ion channels is that they can show selectivity for the type of ion or ions that can diffuse through them. This selectivity is based on the channel diameter, the charged and polar surfaces of the polypeptide subunits that form the channel walls and electrically attract or repel the ions, and the number of water molecules associated with the ions, so-called waters of hydration. For example, some channels, such as potassium channels, allow only potassium ions to pass, whereas others are specific for sodium ions, which are sodium channels. For this reason, two membranes that have the same permeability to potassium, because they have the same number of potassium channels, may nonetheless have quite different permeabilities to sodium if they contain different numbers of sodium channels. Moving forward, integral membrane proteins can span the lipid bilayer. Some of these proteins form ion channels that allow ions to diffuse across the membrane. A single protein may have a conformation resembling that of a donut, with the hole in the middle, providing the channel for ion movement. More often, several polypeptides aggregate, each forming a subunit of the walls of a channel. The diameters of ion channels are very small, only slightly larger than those of the ions that pass through them. The small size of the channels prevents larger molecules from entering or leaving. Let's understand it in detail. Model of an ion channel, composed of 5 polypeptide subunits. Individual amino acids are represented as beads. First, a channel subunit consisting of an integral membrane protein containing 4 transmembrane segments, 1, 2, 3, and 4, each of which has an alpha helical configuration within the membrane, although this model has only 4 transmembrane segments. Some channel proteins have as many as 12. Secondly, the same subunit as shown before, in 3 dimensions within the membrane with the 4 transmembrane helices aggregated together and shown as cylinders. Thirdly, the ion channel consists of 5 of the subunits, as illustrated in D, which form the sides of the channel. Now let's cross-sect it. The helical transmembrane segment 2, which is in light blue color, of each subunit forms each side of the channel opening, the presence of ionized amino acid side chains. Along this region determines the selectivity of the channel to ions. This is aqueous pores of ion channels. Although this model shows the 5 subunits as identical, many ion channels are formed from the aggregation of several different types of subunit polypeptides. Moving forward towards the effects of electrical forces on ion movement. So we have described the direction and magnitude of solute diffusion across a membrane in terms of the solute's concentration difference across the membrane, its solubility in the membrane lipids, the presence of membrane ion channels, and the area of the membrane. When describing the diffusion of ions, because they are charged, one additional factor must be considered, which is the presence of electrical forces acting upon the ions. A separation of electrical charge exists across plasma membranes of all cells. This is known as a membrane potential, the magnitude of which is measured in units of millivolts. The membrane potential provides an electrical force that influences the movement of ions across the membrane. A fundamental principle of physics is that, like charges repel each other, whereas opposite charges attract. For example, if the inside of a cell has a net negative charge with respect to the outside, as is generally true, there will be an electrical force attracting positive ions into the cell and repelling negative ions. Even if no difference in ion concentration existed across the membrane, there would still be a net movement of positive ions into and negative ions out of the cell because of the membrane potential. Consequently, the direction and magnitude of ion fluxes across membranes depend on both the concentration difference and the electrical difference, also called as the membrane potential. These two driving forces are considered together as a single combined electrochemical gradient across a membrane. The two forces that make up the electrochemical gradient may in some cases oppose each other. For example, the membrane potential may be driving potassium in one direction across the membrane, while the concentration difference for potassium is driving these ions in the opposite direction. The net movement of potassium, in this case, would be determined by the relative magnitudes of the two opposing forces, that is, by the electrochemical gradient across the membrane. This was all about this section. In our next section, we will talk about the regulation of diffusion through ion channels.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.